Hello everyone, and welcome to today's webinar, Simulation and Modeling of Bioenergy Conversion from the Atomic to Process Scales. Before we get started, I'd like to go over a few items so you know how to participate in today's event. You're listening in using your computer's speaker system by default. If you would prefer to join over the phone, just select Telephone in the audio pane of your control panel, and the dial-in information will be displayed. You will have the opportunity to submit text questions to today's presenters by typing your questions into the question pane of the control panel. You may send your questions in at any time during the presentation. We will collect these and follow up with you after today's presentation. Our presenters today are Sona Kim, Peter Suzelski, and Jim Parks. Now I'll turn it over to Jim.
Hello everybody, I'm Jim Parks from Oak Ridge National Lab, and I'm happy to present to you today activities we have in computational science related to ChemCatBio and the Bioenergy Technologies Office. We'll be giving a tag team presentation and very much a collaborative presentation and also a collaborative effort in modeling in this program. So as you can see from the title slide, there's quite a few people involved in this, and there are 5 national labs representing this part of the program. So first, we'd like to talk about the Consortium for Computational Physics and Chemistry from a high-level view. We are 5 national labs: Oak Ridge National Lab, Pacific Northwest National Lab, the National Energy Technology Laboratory, the National Renewable Energy Laboratory, and Argonne National Lab. And we work together to perform modeling and simulation to support the many activities in the Bioenergy Technologies Office at the Department of Energy. And today we're going to be specifically talking about our support of the ChemCatBio program, and as we're sharing the info for this ChemCatBio webinar. We also support other activities in the program, such as biopower, biochemical process modeling and simulation, the Feedstock Conversion Interface Consortium, and the Bioprocessing Separations Consortium. Today we'll be talking to you about 3 different areas. This is kind of how we organize our team. We have modeling activities going on at the atomic scale, where we're doing modeling of catalysis at the very molecular level. We also have modeling at the meso scale, where we're actually modeling catalyst particles and the different processes that go on at that scale. And finally, we do modeling at process scale, full reactor systems. And so today we're going to give you some examples of the modeling work that is going on at these 3 different levels. And we'll have Sona Kim talk about the atomic scale, Peter Suzelski talk about mesoscale, and then I'll follow up with process scale modeling. In addition to this webinar, if you'd like to know more about our consortium, you can check out our website, which is www.cpcbiomass.org. So now I'm going to turn it over to Tiana, who will talk about atomic scale modeling.
Thanks so much, Jim. I'm Sona Kim from NREL, and I will present an atomic scale catalyst modeling that we are doing in this part of the CCPC activities. So when we try to guide compositional catalyst design using multiple atomistic toolkits, so starting from using very high-level theory of, for example, QM-DFT DFT method to do the atomic the active site study. And also we add some kind of the confinement to the QMMM study also. And also we expand to include the surrounding medium to using the MD simulation. So when we start, we have some catalyst substrate system, and through the mechanism study, we do the thermodynamic and kinetics study, and including all of these kind of information, we do the screening the catalyst and with the best activity and selectivity catalyst to test in the experiment. This is one the group of the work, and we do that we use a lot of feedback from experimental group and vice versa. So we do the a looping thing for the inside of the CCPC activity. So our ultimate goal for the atomistic simulation team to support the ChemCatBio, first, we want to understand catalyst activity and selectivity as a function of the composition of the catalyst. And also, we want to guide new catalyst synthesis and then work with the AC/SC team inside of the ChemCatBio. And finally, not only the designing the catalyst active site, we also want to optimize the operating conditions like temperature and pressure and etc. How we do the atomistic modeling to achieve these goals? We do the modeling for the both homogeneous and heterogeneous catalysts to understand the reaction chemistry. Also, we also explore the catalytic cycle and with the nature of the active site to understand the structure and function relationships. We also expand to the exploring the molecular scale transfers, for example, using the molecular dynamics simulation. And finally, we also screen the catalyst to guide the new synthesis using the machine learning, linear regression, and DFT. So this is what we want to achieve, the goal to design the performance advantages of catalyst. Here, I want to share a few examples that we are doing right now. So the first example that I want to share with you is develop the bimetallic catalyst for selective carboxylic acid reaction. So along with a lot of different the experimental group collaboration, we tested 3 different surfaces, tin ruthenium and ruthenium and tin oxide on top of the ruthenium. For the tin ruthenium and ruthenium surface, you see a lot of side effects along with the acid and carboxylic acid reduction with high energy barrier. However, when we use the tin oxide surface, this reaction is becoming very selective for the carboxylic acid reduction with a very low activation energy barrier. So this is what we did to rationalize the catalyst stability and also proving the mechanism for this reduction reaction. And then these details we try to evaluate using the experimental team. And also, we try to tune a little bit of the design parameters to improve the catalyst activity and reactivity. And then we tested, from the with the experimental group. Our second example that I want to share is to understand the water deactivation of the metal oxide catalyst. We chose magnesium oxide because this is very well known crystal structure they have. And also, it's relatively a lot simpler than other metal oxides. So our goal is to understand the water deactivation and also with reactivity of the catalyst. So first, we started with the cold adsorption equilibrium phase diagram, and as shown in the left-hand side here, and then to see what is the reaction between the surface and then reactant here. Using those information, we studied the reaction energy differences of the metal oxide. Our target reaction is the azo condensation here. At the same time, not only the reactivity we also studied the water tolerance and also catalyst deactivation. And one of the third example that I want to share is ethanol upgrading over HGSM5 zeolite here. We studied not only the internal of the pore, we also studied the external of the pore, and then to think about what is the effect, critical effect for the enthalpy and entropy contributions to understand the Gibbs free energy here. So you see here in the left-hand side graph, you see some difference between the external and internal branched acid sites, and for the enthalpy and also free energy, and also they can be affect some reactive rate determining step also. Here in the right-hand side, that we tested single branched acid sites versus double branched acid sites, showing the improvement both internal and external pore, and also we try to decrease the temperature, and then you see a lot more more enhancements for both internal and external pore. So we think we just concluded like thermodynamic profiles is like tuned by increasing the breathless size and also lowering the operating temperature. So that is what it is a good kind of point to to the experimental group. So we also, as I mentioned earlier, we also did some MD simulation for this kind of work, and then I want to share micropores versus mesoporous geolite catalysis work used for the diffusion, because diffusion is very important in biomass upgrading, which impacts the co-formation and also protects the selectivity and yields and separations like that. So the first is the top image I'm showing you here is this is the micropores in GSM-5, and then we compare like a paraxylene versus toluene here in this figure. And when you see the toluene case, they reorient a lot more when they pass through the pore to the next adjacent pore. So it diffuses a lot slower than paraxylene here. We also calculate the diffusivity of the co-precursor. Here we use the benzene, naphthalene, and anthracene as an example in both micropore and mesoporous here. And then mesopore, we use the two different mesopore sizes here. And then this is one of the example in benzene diffusion in mesoporous, how they work from mesoporous to the micropore here. And then this is another the motivation we study for the co-formation work. We think the co-formation is also very important inside of geolites and also external surface. So the first example is we ran ethylene, a lot of ethylene here inside of the micropore to see how they form the Foley-N or chain growth here. So they cannot make a lot of like a kind of benzene style of the co-formation here because of the pore size. So we are thinking to move on to the external surface here, and this is very preliminary result. So I'm just showing you the in gas phase how they can form from chain growth and then ring formation and then PAH formation here like this. So now we want to incorporate this work put it mesopore at the external surface of the geolite catalyst and then see how it how the co-formation formed. That is what we have here. So this is what we are in the atomicity modeling work. And then I will hand over to Peter Strzelski for the mesoscale particle modeling.
Thanks, Sona. That was really good. And you did a good job setting up a lot of the stuff that I want to talk about. So I guess I'll just motivate mesoscale modeling. It's kind of abstract, I guess, at first if you're not used to hearing about it. So whenever you go and measure experimentally the kinetics, by a bench scale or small reactor scale experiment, really the observed reaction rate is a function of a lot of different things. So there's the intrinsic reaction kinetics that are really how quickly a chemical reaction forms once the reactant has reached an active site, how quickly that reaction can proceed. So that's what we'll call the intrinsic reaction kinetics. And then also that's delayed by the time at which it takes for a reactant molecule to diffuse into a catalyst particle and reach an active site. So that's a delay that will also manifest in the observed reaction kinetics. And then finally, there's so that's really a diffusion process, and Sona did a good job of talking about the importance of intraparticle diffusion. There's also extraparticle mass transport limitations. For example, reactants and products diffusing through a boundary layer that sets up around a catalyst when it's in a velocity field, and we're doing a lot of calculations recently that highlight the importance of that process. So all these things together play into what you're measuring experimentally, and so mesoscale modeling is one way that we can handle these things explicitly, and it's a great tool we found for interpreting experimental results. So I'll start by showing you what a zeolite catalyst particle looks like. This is an XCT scan, and you can see kind of the porosity of this particle. So that's one factor we can consider. And then if you take a cross section of this in the TEM, you can see the porosity, where how the catalytic sites are kind of bound together with binder. And then if you zoom way in on that, eventually you can see the microporous structure of the zeolite where the active sites actually reside. And so transport through each one of these length scales is significant. And then the other thing it's important to remember is that, so we've got down here just an optical micrograph of a catalyst particle that's partially deactivated from a reactor here at NREL. And so you can see how it sort of deactivates from the outside in. So at any given time, a fraction of the catalyst sites are active and a fraction of them are deactivated. And it's really a function of transport throughout the catalyst particle and the residence time of a catalyst particle within a reactor. And so all of these things provide opportunities to optimize not only the catalyst design, but also the operating conditions to provide the most optimal interaction between catalyst particles and reactants. So our approach to doing this is we use very detailed characterization of catalyst particles to measure directly the porosity, and this is again with image analysis that we perform on XET reconstructions. And in this particular example, the catalyst particles that we were working with have two pretty distinct porosity domains, where the outer region has much more void space and the inner region is a lot much more dense. And so as you might imagine, transport through these different regimes is quite different, and we're able to, we can model that explicitly, and also we can use approximations for transport in porous media, which actually reduce the computational load substantially. And so there's a kind of a finite element domain of finite element model here showing how we model this, and then there's a few conservation statements there at the bottom that we use to solve this in a finite element type format, and I won't belabor the math today. So this is for a Z-like catalyst particle, and I think Jim will talk more about later how we use these types of simulations to extract kinetics that are independent of transport phenomena. But I just want to show you what one of these simulations looks like. And so when I start this movie, you can see the experimental data that are plotted on the left, and then we'll plot the line of what the simulation predicts through those data, and then on the right is just visualizing the coke formation and product evolution that's inside one of these particles. So, and I should say that these data were actually optimized. The simulation was in fact optimized for the simulation results. So this is an example of where we used the mesoscale simulation to extract intrinsic kinetics. So then as this movie plays, you can see we're able to predict spatially and temporally coke formation within the catalyst particle. And I guess the other interesting thing that we learned from this simulation is that if you look at the very last time step, which is where the movie stopped here, you still have a lot of product that's trapped within the catalyst particle. And so in a reactor scenario, this catalyst particle would then go on to a regeneration cycle where all the coke is burned off. And so you can also estimate how much of your desired product you're essentially forfeiting at that point, which is, again, another opportunity to optimize your operating conditions at the reactor scale. So we also, like I mentioned, this work is tightly coupled to advanced catalyst characterization, and we're working with the advanced catalyst synthesis and characterization team, particularly Kinga Unokic from Oak Ridge National Lab, who uses advanced microscopy and chemical imaging to help us validate, for example, our predictions of coke formation in these catalyst particles. And so finally, I just want to cover another example of this where we're looking at supported catalysts. And so this is in collaboration with our experimental partners at PNNL, particularly the group of Rob Daigle. They're using SPA mesoporous silica to support a catalytic system that converts ethanol to butadiene. And this is a nice platform in terms of modeling and also in terms of catalyst design, because it's quite a tunable system. And so the mesoscale really covers from the atomic scale, and it builds a bridge up to the reactor scale that Jim's going to talk about later. And we're able to handle all the important transport phenomena that occur essentially in that regime. So in this project, we started at the atomic scale, and we did molecular diffusion simulations, very similar to what Sona Kim just described to you in the previous section, where we were able to compute diffusion coefficients for reactants and products within the mesoporous structure. We then take that and tessellate it into a larger domain, and then we apply the effective diffusion coefficients for the different locations, and then we're able to do finite element diffusion calculations over a larger scale, and then we can incorporate that again into a particle scale and finally into a reactor scale. So I'll walk you through sort of each one of those scales here in the next few slides. So these are the diffusion calculations where we we're able to calculate diffusion coefficients for reactants and products in the different kind of regimes that within these catalysts. So within the necks that connect these larger cages in the SBA16 structure. And I guess the reason why that's significant is you see a lot more surface effects where the molecules are confined within those narrow regions, and the diffusion within the larger cages actually begins to look more like bulk diffusion. But we're able to compute directly what transport through those different regimes would look like. So then what we do is we take those diffusion coefficients and we apply them in a finite element simulation. So in the cage domain, we use the cage diffusion coefficient, and in the pore domain, we use the pore diffusion coefficient. And then from there, we're able to apply concentration gradients across this, and now we're doing finite element calculations. And from there, we're able to compute effective diffusion coefficients that take into account both regimes, and then apply that, which will allow us to do reaction diffusion simulations at much larger length scales. That's great. That's what we need, because the experimental data that we're using was collected in a packed bed, and so so now that we have effective diffusion coefficients that essentially account for molecular level and mesoscale transport, we're able to overlay that with chemical reaction kinetics and fit directly compare directly against experimental data, and we can get very nice agreement, and we can also extract chemical reaction kinetics for different systems. So we're solving conservation equations again for, in this case, just mass and momentum. So we assume this to be isothermal, although it's easy to incorporate heat transfer if that's significant. In many cases, it is. And then we use again approximations for transport in porous media to account for transport not only through the gaps between particles in the packed bed, but also, as I mentioned, through all the different porosity regimes within these catalyst particles. Okay, and then what's nice about this is that once we have a parameterized simulation, we can begin to do sensitivity analysis for parameters that are easy to control across different length scales. So one nice thing about SBA16 is you can tune the cage size, you can tune the pore size that connects the cages. That's sort of at the nanoscale. And so we can do parameter sweeps over that to see how that affects the performance of the catalyst. And then, so essentially we asked our experimental partners, what catalyst attributes are easy for you guys to tune? And they told us particle size, and they told us changing the pore size. And so we do parametric sweeps over that, and we're able to provide them predictions of how changing those things is going to impact the performance of their system. And this is kind of a nice, tidy analysis, because at the end of the day, we can provide them very actionable predictions. And so what's shown there in the bottom right is that it's as simple as reducing the catalyst particle size and increasing the pore size. Our models predict will result in improved catalyst activity lifetimes. And so those are experiments that are going to be performed hopefully next year. Yeah, I think that's all I want to say. And now I'll pass it over to Jim.
Thank you, Peter. So we'll finish by talking about the process scale modeling. And of course, as you can see, there's been a lot of research in supporting the catalyst innovations in ChemCatBio, and then understanding the phenomena at particle scale as well. And we also needed to do this at the process scale. Our ultimate goal, of course, in ChemCatBio is to support the bioenergy industry, and they have reactors in the private sector. And we're also working towards that goal to support those efforts. And we have reactors in the National Lab system as well. And we're modeling those reactors to support the scale-up of catalysts from the innovations that occur in ChemCatBio all the way to demonstrating them in process scale. So a lot of the process scale modeling is done with computational flow dynamics, so multi-phase flow. And what you'll see here is model on the left of a reactor called the R-Cube catalytic upgrading reactor. This is an entrained flow reactor where the catalyst particles enter the bottom and mix with pyrolysis vapors. And then as you go up the column, those catalysts then help to convert those pyrolysis vapors into hydrocarbons that are more appropriate for feeding into our fuel infrastructure in the United States. So you can see on the right the actual reactor, which is at the National Renewable Energy Laboratory. And so we work very closely with those experimentalists on that team, where they're taking validation data for our models. And then ultimately we want to have this computational and experimental data to compare and frame the complete picture of the catalytic upgrading that's occurring. And by the way, this type of model is done with MFIX, the multi-phase flow with interphase exchange, which is a CFD type code that was developed by NREL as part of our program. And so this is an open source code, and so whatever tools we develop and models we develop are directly portable to anybody. So in terms of the reactors that we're modeling in the program, we actually have 3 different size reactors. This one is a 2-inch fluidized bed reactor, then we have a Davison circulating riser, and then finally the R-cubed upgrading system, which you saw on the previous slide. Important thing to note about these reactors is they operate at very different scales in terms of the biomass going into the reactors. And so you can see the scales in terms of kilograms per hour demonstrated here. They also have different relevance relative to the purpose. The smaller reactors, the benchtop reactor, which is a 2-inch fluidized bed reactor, is really one of the first catalysts that you'll see the new catalyst innovations tested on. And so it's important to understand those early results, and so we're modeling that type of a system. Then the larger one is actually getting closer to something that's more relevant to industry and relevant to the verification program that goes on in our research program. The Davison circulating riser is also a relevance to industry, because industry, and particularly WR Grace, uses this system to actually provide performance information for their catalysts. So there's a lot of different reasons to study these different reactors at different sizes. An important aspect that the process scale models of these reactors gives us is the actual residence time distributions of the catalyst in the reactor. And so here we show on the left, we show MFIX simulations of a Boeing bed reactor. This is the 2-inch fluidized bed reactor. And we inserted a tracer to study the flow of new catalysts going into the reactor. And then the Boeing bed, those catalysts mix, and then actually the catalysts exit out of the reactor. On the right, you see the R-cube reactor, which is that entrained flow system. So these 2 very different types of reactors have very different residence times. For the 2-inch fluidized bed reactor, we have over 800 seconds of average residence time, whereas in the riser, we're looking at only about 139 seconds. So very different residence times, of course, will lead to very different performance, and then also the catalyst particle sizes vary as well. So these are the important aspects in terms of the process that we need to capture with these models and are able to do so. And then Peter talked to you about a lot of the work at the meso scale. And one exciting thing that we've been doing here recently in the program is developing kinetics specific to these complex bioenergy processes. And in particular, the catalyst and process that we've been studying is the catalytic fast pyrolysis, and looking at the upgrading of those pyrolysis vapors with a ZSM5 catalyst. And the approach we're using is to use a lump sum type approach to characterize the kinetics of the different reactions going on. It's important to note that there's a lot of activity out there studying kinetics, and particularly at the universities, for instance, at Northwestern University and the University of Delaware, Professors Brauveld and Klein, respectively, at those institutions are studying microkinetics. We're very much in support of that effort, and hopefully would like to include those kinetics in our models at some day. But for the time being, we're using a simpler scheme that does capture fully the products that are coming out that we can measure, and then we can actually get these kinetic rates. So this has been done in close collaboration with experimentalists in the ChemCatBio program, and you can see some of the kinetic rates here on this slide. Peter mentioned to you a little bit about this process, and this is a little bit more detail on this slide and the collaboration with the experimentalists to get these kinetic informations. And so we've been working closely with experimentalists who have been running the catalyst in a spouted bed reactor and then detecting the products with molecular beam mass spec detector. And then with feedback of our models that Peter developed, we've been able to come up with these kinetic schemes. So at this point, we're actually using those kinetics in our models. And so this shows a model of the R-cube reactor, but showing the reactants, the pyrolysis vapors, along with the products groups that we've determined for this kinetic scheme. And you can see that we get a different distribution of those products as you go up the reactor. But we're at the point now where we can actually run these models and predict the performance for these different products. And we're now working on validating the models together with our experimentalists. Some of the validation of the models is shown here. This is actually just the hydrodynamics. And in the next coming months, we expect to get our initial results on the actual upgrading itself. But for now, we've been looking at pressure differences in the reactor and comparing that to what's predicted in our models based on the hydrodynamics, and have come up with pretty good agreement there. So we're really excited about the upcoming validation experiments that will be done with the actual pyrolysis oil. So just to summarize, I'd like to point out that the modeling that we do in this program is very much in support of ChemCatBio. We see ourselves as an enabler to enable success in the innovation of new catalysts, and then also the understanding of the phenomena that can lead to getting those catalysts working in true processes that would be of interest to the bioenergy industry. As we mentioned, and as you saw, we've got activities that actually cover a very wide range of scale. Sienna talked about the atomic scale modeling, Peter talked about the mesoscale, and then I followed up with our activities at the process scale. Again, we thank the experimentalists in the program and also our industry advisors that have been helping us to get this work done. And we're excited about the kinetics that are coming with this and our application of those, and we look forward to more experimental validation of that as we go forward in the program. And finally, I'd just like to leave the last slide up with the many names of the researchers at the 5 national labs that have been contributing to this model program, and also like to thank the Department of Energy Bioenergy Technologies Office, in particular, Chairman Leong, Trevor Smith, and Kevin Craig. And then also like to give a shout out to our industry advisory panel. We utilize their input to help steer us into catalysts and processes of studies that are very relevant to the bioenergy community, and so we appreciate their help in doing that. And I guess now we'll get to take any questions.
Great, thank you, Jim, and thank you, Sona and Peter. It looks like we do have a bit of time for questions today, and as a reminder, you can still submit questions through the question pane in your attendee control panel. Our first question here says, Peter, are you using dual porosity porous flow model to account for interparticle porosity and interparticle packing porosity at the same time?
Yeah, so that's a great question. I guess effectively that's what we're doing. So essentially we build up the transport model like an onion, so to speak. And so first we compute effective porosities in each of the nanoscale domains of the catalyst. And then we apply that to a finite element model where we can account for effective transport throughout a catalyst particle. So that's intraparticle diffusion. Okay, and so then we essentially couple that to it's basically a Thiele modulus sort of Darcy's law type approximation that accounts for. So now we have an effective diffusivity within a catalyst particle. Now we have to account for transport between catalyst particles in a packed bed. And that's done with another type of approximation. So yeah, so that's a good question. Whoever asked that, we are accounting for those different types of transport phenomena that arise at different length and time scales.
Great, thank you. And that person did say thanks.
Great.
So we do have another question here. It says, a more general question: does CCPC consider the particle level modeling for coupled transport generation expulsion of biofluids within biomass particles? This could be critical for estimations of effective conversion kinetics that goes into the macroscopic reactive transport model.
Yeah, so again, that question I think is towards actually conversion of the biomass particles in the initial deconstruction step. That's how I'm interpreting it, at least, and so I think that's kind of. So, can you repeat that one time?
Yep, it's right here.
Okay. Yeah, so we have very, very detailed biomass particle thermochemical conversion models, and we didn't cover those today. You know, we've been working on those for actually longer than we have the catalysis models, and we just published a paper in Energy and Fuels. Actually, we got the cover. I'd be happy to send it to whoever asked that question, where we do detailed transport phenomena coupled to thermochemical reactions within biomass particles, and we account for the microstructure of those particles. I think this question was kind of focused on you know, the ejection of vapors and products as those biomass particles convert. And so we handle that pretty well. We make assumptions where we can, but I should say we were able to predict the conversion yields coming out of a reactor at NREL within 0.1% of the experimental measurement. So that's probably better than the experimental error, but we can make very, very accurate predictions.
Great. So follow up on that, it says, Peter, it's hi from INL. Please send me the paper you just mentioned.
Sure.
Great. So it looks like we don't have any other questions at this time. I'll just give everyone another minute to go ahead and submit any questions through the question pane of your control panel. We can also follow up on any other questions you might have after the webinar today.
While you're doing that, I'll just add that to what Peter mentioned about the thermochemical models of biomass particles. You can read more about that in the paper, and we have a list of our publications on our website. So feel free to go check out the website and see that. You probably also see some examples of the models of the biomass particles that Peter was talking about.
Great, thanks. It looks like we don't have any other questions at this time. So thank you to all of our presenters, and thank you everyone for attending today's webinar, Simulation and Modeling of Bioenergy Conversion from the Atomic to Process Scales. You will receive a follow-up email within the next couple weeks with a link to view a recording of today's webinar. Thank you for joining us today, and have a great rest of your day.